Right. Well, I guess the first thing I should tell you is, if you notice, the title of this talk is in English, and we all think that it would be a good thing for you to have more teaching in English during your master's course. And it's, at the moment, this is kind of just a vague wish. So you don't have very many lectures in English, either in BBRT or in SCMV. But it's always good to start somewhere, right? So I'll be speaking in English for an hour and a half. And the rest of the slides have French text. Okay, so at least if what I say isn't very clear, you should be able to, understand what I'm talking about from the text. On the other hand, right, if I start speaking too quickly and you can't follow me, then you should say something, all right, or make a big sign, like go like this, whoa, slow down, like that, okay? Otherwise, personally, I'm not going to notice. Okay, so if I'm going too fast, please tell me. Okay, so we have 1.5 hours to think about antiviral immunity. So if you think about a simple self-limiting acute viral infection, what you generally have is you get infection, you get a peak of virus replication. It's going to be followed within a couple of days by the innate immune response. So you'll have interferon is produced, you'll have fever, you'll have inflammation locally at the site that's infected. And this will be followed closely by the natural killer cells, the NK cell response, so innate effector cells that kill infected cells. Then you get the adaptive immune response, which will come slightly afterwards. So maybe, I don't know, within about a week or so, you might get CTL. And then after maybe from 2 weeks onwards, you start to see specific IgG, IgM and IgG. So you get the humoral response. So you have this kind of cascade of the innate response as first. It's going to be essential to have this innate response. Otherwise, your adaptive immune response is not going to happen. Now, what I want to do for the rest of the time is I'm not going to talk really about dendritic cells very much, just slightly. I'm going to mention them in passing. But I'm not going to talk about T helper cells or cytokine networks. It's not because these things aren't important. They're essential to coordinate the adaptive immune response. But my focus today is going to be on the effector mechanisms that directly prevent or limit virus replication. So I'm going to focus on the things that are on this slide. Okay, the interferon response. So why, in fact, why do cells become resistant to virus infection after they're treated by interferon? So we'll look at a bit about the molecular mechanisms involved in that. And for NK and CTL, I'm going to lump them together as cytotoxic cells. And really the focus is going to be on how do these effector cells recognize infected cells, and how do viruses escape from NK and CTL lysis. And I'll be doing the same kind of thing with antibodies. What are the characteristics of neutralizing antibodies? And why don't we have a good neutralizing response against all viruses? Because that's going to be important for vaccines. Okay, so getting back to the beginning, we'll start off with the innate response. And really, I want to be looking at individual cells. How do cells individually respond to viral infection? It's not really part of the immune system. It's kind of like virus-cell interactions, but you could also think of it as being, the first line of the innate immune response. The first problem that cells have in really responding to a virus infection is just knowing that you're infected in the first place. How does a cell know when a virus is present? And it's not a simple question, because viruses are made by other cells in the body. So they're made out of the same biochemical material. lipids in viruses, they're the same as lipids in cells. Viral proteins, they're the same as like cellular proteins. So it's not like a bacterial infection or fungal infection where you can have very specific molecules produced by the pathogen that can be recognized by host cell innate receptors. So that's the first problem for detecting viral infection. And most of the first line detection of viruses depends on detecting viral nucleic acids. And there's two types of pattern recognition receptors involved in this. So the first are like the toll-like receptors, 3, 7, and 9, that can detect the presence of extracellular nucleic acids. Now these receptors, they're not on the plasma membrane, they're on the endosomal membrane. And that's important because generally you don't have nucleic acid floating around in this extracellular fluid. Right? You've got a lot of DNAs and RNAs in plasma and extracellular fluid. Free nucleic acids get degraded very quickly in the body. However, virus particles, of course, resist this because the nucleic acid is inside the capsid. However, when these particles are taken up by dendritic cells or other cells of the immune system, then the particles are degraded in the endolysosome, and this liberates nucleic acid. Okay? And then these combine to toll-like receptors, and they'll activate those dendritic cells. So these kind of receptors here are on cells of the immune system. It's the way that your immune system is going to detect the presence of viruses. So the cells that are activated here are not necessarily infected, okay? Not productively infected cells. These are cells that are detecting viruses outside of the cell, taking them up and becoming activated. So that's one class of pattern recognition receptors involved in identifying virus infection. And the other class are these whole family of cytoplasmic receptors. So these are involved in, when an infected cell has virus replication going on inside it, it recognizes I'm infected by the activation of some of these receptors. So there are a different number of receptors for DNA. Okay, in a normal cell, your DNA is inside the nucleus or in the mitochondria. You shouldn't have any DNA in the cytosol. So if you've got DNA there, something is wrong. Okay, so that's basically what these ones are for. These MDA5 and RIG1 that recognize specific RNA structures that are not usually found in a non-infected cell. Okay, so double-stranded RNA, not generally found in the cytosol, but it's very often formed by RNA viruses as a replication intermediate. Okay, double-stranded RNA viruses, okay, they already have double-stranded RNA. Single-stranded RNA viruses, they've got to copy their genome, so they make a complementary strand. They've got to have double-stranded RNA at some point. And that's why this one is specific for the presence of viral RNA. This one, RIG1, it recommends the 5' triphosphate RNA. So normally, the 5' end of tRNA or ribosomal RNA is like buried inside the 3D structure of the RNA. Messenger RNA in the cytosol has got a 5' cap, so it doesn't have a free triphosphate group on the end of it. So often viral RNAs don't have a 5' cap. So they're detected specifically by RIG1. So that's what these pattern recognition receptors are doing. So what's the consequence of their activation? Well, both of them are involved in the secretion of type 1 interferons. So that's interferon alpha, interferon beta. So dendritic cells that express these toll-like receptors, when they're activated, they'll express alpha interferons. And infected cells, once they have one of these receptors that is activated, they'll express beta interferon. Now, in addition to that, so toll-like receptor stimulation in DC will mature dendritic cells. It's going to play an important role in activating the adaptive response, okay? Because these cells will start presenting antigen to T cells, activate them, and that's how you're going to start getting a CTL response. Now, another consequence of the activation of some of these cytoplasmic receptors is programmed cell death. So these are really like the two choices that an infected cell has got once it detects that it's been infected. Either the infected cell can try and secrete interferon, and that will make the neighboring cells resistant to subsequent infection. Or if that doesn't work, or even at the same time perhaps, the infected cell is going to try and kill itself and start cellular suicide. Okay, so the questions I want to look at now are, so we've got all these kind of like bricks inside these cells. They become resistant to viral infection. So why? Why is that? What happens to cells that make them so resistant? Okay, so that's the first question. Then afterwards we'll look at this part. Okay, what are the different pathways of programmed cell death? How does virus infection activate them? And how do viruses avoid this kind of defense mechanism? Okay, so on the first point, okay, how does interferon make cells resistant to virus infection? Well, it does that by inducing the expression of host cell restriction factors. Okay, so cells that are stimulated by interferon will transcribe maybe a couple of hundred interferon stimulated genes, and many of these are restriction factors. So these will block different steps of the virus infection. So they may have a constitutive expression at a low level. They're not normally expressed at a high level in most cells. Their functions are to block different steps of virus infection. And the reason why viruses can still infect us is that they often express accessory genes that are going to interfere and handicap these host cell restriction factors. And as we'll see at the end, this results in a kind of evolutionary arms race between viruses and hosts. And it's one of the important parts of the species barrier that prevents us getting infected by a lot of animal viruses. Now, so this slide is from a review from 2013, and I thought I would kind of just go through each of these restriction factors and explain what they are. But I'm not going to talk about RNase L and ADAR1 because generally Jacques de Seyek covers these effectors in his lecture. And Viperin, I checked just last week, still not entirely clear to me what the mechanism really is of this effector molecule. So I'm kind of waiting till I understand how it works myself before trying to explain it to anyone else. Okay, but so we have this list of restriction factors. How on earth did we find out about these things? Okay, how do we know that they exist? Well, one of the approaches that's been used is trying to understand why some cells are permissive for viral infection. So that means the virus can complete its replication cycle in this cell type, whereas other cells from the same organism are non-permissive. Now, in non-permissive cells, the virus might be able to bind the cell, so the receptors present, it might be able to enter the cell. So early steps are possible, but somehow it can't replicate. Okay, so once you see that, there are two possible explanations. Either your cell B lacks an essential cofactor for infection, and that's why it's non-permissive. Or maybe B expresses a restriction factor. Okay, and it's the absence of the restriction factor in this cell line A that allows the virus to replicate. And there's a really quite easy way to determine which of these two cases is correct. and that is by producing a heterokaryon. So if you fuse these two cell types, you get a heterokaryon. And then if you try and infect this with the virus, if your heterokaryon is still non-permissive, that means that you've got a restriction factor. So having a restriction factor is it's a dominant phenotype. Okay? Does everybody understand why that's the case? Yeah. So a lot of these restriction factors were discovered by looking at this type of system. And then once you've defined that, then you can say, okay, what genes have I got that are expressed in this cell and they're not in this cell? And you can try and clone them and see whether one of them is a restriction factor. Or you can try a kind of expression cloning approach, make a cDNA bank from this cell line and screen it by transfecting them into the permissive cell line. And then one of these clones will make your permissive cell resistant. And that clone codes for the gene that is the restriction factor. Okay, so that's how people have approached this kind of question. Right, so let's have a look at some specific examples. TRIM5 alpha. Okay, so this was originally discovered because people found that cells from the rhesus macaque are not permissive for infection by HIV-1, even though the virus can bind to the cells and enter them. And then, of course, human cells are permissive, so it's possible to try and to clone the gene from the rhesus macaque that was responsible for this restriction. And it turned out to be this molecule, TRIM5A. So humans have an equivalent. Okay, so we have human TRIM5A. It's not effective against HIV. So that's why we are susceptible to this virus. But it does prevent us getting infected by murine retroviruses. So in the past, it has been very good at protecting us from being colonized by certain types of retroviruses. So how does it work? Well, the TRIM5A protein seems to form these kind of trimers on first, okay? And then they bind onto the capsid of the retrovirus just after entry, but before reverse transcription. There's a very early step in virus replication that's getting blocked. Now, another thing that is interesting here is when you look at TRIM5A genes in primates, they're quite diverse. There's quite a bit of sequence polymorphism in these genes. And even, in fact, in the human population, there are some polymorphisms. So there are some single nucleotide polymorphisms that change 1 or 2 amino acids in the human TRIM5A protein. And there are some genotypes that are associated with increased resistance to HIV infection. So, you know, if we didn't have any treatment for HIV and we had a huge epidemic where we had a lot of people getting infected, then those individuals that have got the genotype that allows them to resist the infection here would become more frequent in the population. Okay. So viruses, how do they respond to a restriction by TRIM5A? Well, they mutate the capsid proteins so that TRIM5A can't bind anymore. Okay, so that's a pretty simple mechanism of resistance. Okay, so the next one we're going to look at is interferon-induced transmembrane proteins, IFITM. So they were identified not very long ago in a study that was trying to look for host cell cofactors for influenza infection. So their experimental design is like this. They've got cells that are permissive for influenza infection. And they're going to get a whole bank of siRNA and knock down all the genes that exist in these cells. So they have a large-scale screening program. So they knock down all the genes from this pool of siRNA. Then they infect the cells with influenza, and they test, okay, which siRNA cause can inhibit infection. So if your siRNA inhibits infection, then that's knocking down a host cell cofactor. But they also found some siRNAs that increased the percentage, the infection of cells. Okay, so then there is, okay, what is happening here is we've got an siRNA that's knocking down a restriction factor. So they thought that was pretty interesting. Okay, so they had a look at the gene, and that's turned out to be this thing, IFITM3. So they found that when they knocked this down, then they increased the replication of influenza virus. So then they had a look, and they found that it's part of a multi-protein family, and different members of this family can inhibit the entry of different envelope viruses. So what are these things doing? So IFITM1 is in the plasma membrane, and it's also in early endosomes. IFITM2 and 3 are in late endosomes or lysosomes. So IFITM1 inhibits the entry of envelope viruses that can fuse their membrane directly with the plasma membrane or in early endosomes. They don't need reduced pH to elicit fusion. Whereas IFITM2 and 3, they're active against viruses that will enter by fusion after acidification of the late endolysosome. Okay. So once again, it's an early step of virus replication that's being inhibited. So how does this work? Okay, so this is a review from 2 years ago, and there were still different models that were being considered. So one of the ideas was that, okay, for viruses to fuse, to start fusion with the host cell membrane, first step is the interaction between the viral attachment glycoprotein, the host cell receptor. Then often you need a co-receptor to be involved. So you need to form this complex between the attachment glycoprotein, the receptor, and the co-receptor. So maybe IFITM was inhibiting or blocking the fluidity of the membrane and inhibiting the recruitment of the co-receptor. Another idea, quite similar, was that host virus and host cell membrane fusion doesn't occur just when you've got one virus glycoprotein engaged on the surface of the cell. You need several of them all together at the same just exactly the same place where you have contact between the host cell and the virus envelope. So another idea is, so maybe IFITM is inhibiting this lateral movement of receptor and virus glycoprotein complexes in the membrane and prevents the formation of this kind of complex. And the last model was, no, maybe that's wrong. Maybe what IFITM is doing is just preventing the fusion of the two membranes by changing the physicochemical properties of the host cell membrane. So I checked on this last week, and in fact, at least for influenza virus, this model seems to be correct. Okay, so IFITM doesn't seem to prevent the early steps in membrane fusion because this hemifusion state is formed, but you don't get the formation of a complete pore, which is essential for virus entry. So C here seems to be the mechanism. Okay, so now we're going to go on to the APOBEC proteins. And once again, it all starts with the observation that some cell lines are permissive, some human cell lines are permissive for HIV replication, this time for viruses that are deleted for an accessory gene called VIF. And other cell lines are non-permissive. So the idea was, okay, let's clone the gene that's responsible for the restriction of these viruses. And that turned out to be APOBEC3G. Now, how does that work? Okay, so in cells that are infected with a virus that doesn't have the VIF protein, APOBEC3G is packaged into the virion that's released. When this virion comes and infects a new cell, that's when APOBEC3G is going to become active, and it will mutate the viral cDNA that is during reverse transcription. So Gs will be mutated. Okay, we'll see exactly how this works in the next slide. So cytidines will be deaminated. There's two consequences. Either the whole virus genome will be degraded, so the cell doesn't get infected, or the genome will be reverse transcribed, but there'll be a lot of mutations. So you'll have a defective genome that is integrated into the next infected cell. So APOBEC 3G is an interesting one because it's a restriction factor. It doesn't protect the first cell that's infected. It protects the next cell along in the chain of transmission of the virus from one cell to the next. So what does VIF do? Well, VIF will prevent APOBEC 3G from being incorporated into the virion. So then when these cells come and infect the next, when these viruses infect the next cell, then reverse transcription will carry on without any interference from APOBEC 3G. So just a little bit more detail about what exactly APOBEC 3G does. So it's an enzyme, and it catalyzes the deamination of cytidine into uridine. So what's the consequences for HIV replication? Okay, so HIV is a retrovirus, the genome's RNA, and it's going to transcribe it into DNA shortly after virus entry. So this RNA sequence will be reverse transcribed into this one. So this G here will be transcribed into a C in the complementary DNA strand. So if APOBEC 3G gets a hold of this DNA, this cytidine will be deaminated and it will turn into a uridine. And then the HIV reverse transcriptase will just read this and copy it into and put an adenine next to it. So the net effect is you've got a G to A mutation in the genome. And APOBEC 3G is preferential for this GG dinucleotide. That's very interesting because UGG, this codes for tryptophan. If it's mutated, it's going to be mutated into a stop codon. And that's very tricky for viruses to escape from because UGG, this is the only codon where you don't have any redundancy in the genetic code. Only one codon codes for tryptophan. So the virus can't mutate its way out of this one. Okay? If it's got a tryptophan somewhere in the protein that's necessary, it's got to code for it by this codon. And APOBEC 3G will turn these into stop codons. Okay, so that's why the genome is going to be inactivated. So APOBEC 3G, like TRIM5 alpha, it's polymorphic in primates. And so some simian APOBEC 3G is very more active against HIV than human APOBEC 3G. Right? So if you mutate human APOBEC 3G, it can become much more effective. So why does that occur? Well, it's because simian APOBEC 3G is not bound by HIV VIF. Okay, carrying on along in this story, SAMHD1 is another one that's active against retroviruses. And this time, HIV1 does not encode for an accessory protein that inhibits SAMHD1, but HIV2 does have does block this restriction factor. So this restriction factor is expressed in myeloid lineage cells, macrophages, and it's the reason why HIV1 does not replicate very well in macrophages. And the way it was cloned was a biochemical approach to see what macrophage proteins can bind to the HIV2 VPX protein. Okay, so what does it do, SAMHD1? This time it's got quite a simple mechanism of action. It gets deoxynucleotide triphosphates and just dephosphorylates them. Okay, so DNTPs, they're required for retroviruses for their reverse transcriptase. So if you don't have any DNTPs inside the cell, well, the virus can't make its copy of genome into DNA. So that's the model for for the action of SAM-HD1. Some people say it's not quite so simple. That there's other types. There are other aspects to the restriction of virus replication by SAM-HD1. So you might say, okay, this is true for retroviruses. How about DNA viruses? In fact, SAM-HD1 also restricts replication of herpes simplex virus in macrophages, because DNA viruses also need dNTPs. And non-dividing cells don't express a lot dNTPs, right? Okay, last one, and then we can kind of skip to something else. So tethering, BST2. Another one, it was identified by looking at restriction factors for HIV. So this time it's the accessory gene VPU. And if you delete this gene in HIV-1, then you notice that the cells aren't efficiently released from the surface. Viruses aren't efficiently released from the surface of an infected cell. So once again, if you make heterokaryons out of permissive and non-permissive cells, this was found to be a dominant phenotype because VPU was still necessary. So we're looking for a restriction factor. And then eventually it was cloned and identified. So what is this thing, tethering BST2? It's a rather strange looking transmembrane protein. It's got one transmembrane domain here at the N-terminus, and it's also got a GPI anchor. So this is in the cytosolic phase, this is on the outside. And it's a bit unusual in that I don't know if there are any other proteins that have got both these types of anchors. I think BST2 is the only one. So what it seems to do is that it seems to bind virions on the surface of the cell and make a kind of bridge, and it prevents them from being released. Okay, and it seems to be this coil-coil domain, which is just holding the virus particles onto the surface of the cell. Okay, so once this has been discovered, then it was found to be involved in the resistance of cells against different envelope viruses. So retroviruses, filoviridae like Ebola virus, paramyxoviridae, so this is going to be like measles or something, rabies, herpes viruses, their release can all be inhibited by tethering BST2. However, all these viruses can infect us and replicate in our cells. So that means that they all express proteins that can block the action of tethering. So in HIV-1 it's VP2, which blocks this activity. In HIV-2 it's the envelope glycoprotein, which will lead to tethering's degradation. In SIV it's a NEF protein. And in the Kaposi sarcoma herpes virus, it's another accessory protein. Okay, so all these viruses have got some way to inhibit BST2. HTLV1, it's another human retrovirus, doesn't possess VPU or any equivalent, so it just stays stuck on the outside of the infected cell. There's not very much liberation of free virus in HTLV1 infection. Mostly it's cell associated. And that's why, because it doesn't inhibit tethering. All right, so just to kind of summarize this part, okay? The reason why we're not falling down dead and getting killed by any old virus that comes and infects us is because we have a kind of balance between host cell restriction factors that are going to prevent some kind of step of virus replication. However, viruses have got antagonistic proteins that are going to bind to our restriction factors and induce their degradation. Now, if we have a lot of people in the population being infected and perhaps even killed or made ill by these viruses, this exerts a selection pressure on the human population, on the host population, and it's going to select for mutant forms of the restriction factor that aren't inhibited by the virus antagonist proteins. So these restriction factors then become dominant in the population. And of course, that means that you have a new selection pressure against a virus. So virus mutants that express variants of these antagonist proteins are going to be favored, and they emerge, and then they can inhibit the host cell restriction factors. So we've got one of these kind of evolutionary arms races between viruses and hosts, and it's played out in the genotypes of host cell restriction factors and viral accessory proteins. So the result is a kind of equilibrium. Viruses can still get to infect us, but they don't kill us because the accessory proteins that we have are still at least partially active. And when you have transmission of viruses from other animals, there are two possible results. One is that our restriction factors inhibit them completely. They just don't infect us. The other result is that our restriction factors are totally useless, and you can get a very severely pathological infection, or very severe pathology associated with infection. So that's the kind of thing you have with rabies, Ebola virus, zoonoses that are very, very highly pathogenic. Okay, so that was the first part. Okay, the interferon response works by inducing these restriction factors that can inhibit multiple steps of virus replication. So if that doesn't work, the other thing cells can do is to kill themselves, commit suicide. Now, the one that everybody knows is apoptosis, but that's not the only mechanism of programmed cell death. So in apoptosis, you have activation of initiator caspases, often involvement of the mitochondria, and this forms the apoptosome, and that's going to activate effector caspases like caspase 3. Another mechanism of cell death is necroptosis. So cell death receptors can also activate RIP1 and RIP3 kinases. They have to associate together to form this necrosome. That will also activate the mitochondria to produce reactive oxygen species, and that's what's going to kill the cell. Now, RIP1 and RIP3 are substrates for caspase 8. So caspase 8 will cut these proteins and inhibit them. So necroptosis will only occur when apoptosis is inhibited. So it's a kind of backup mechanism. Okay, if you've got something that inhibits apoptosis, the cell can still die by necroptosis. And finally, pyroptosis. This one is dependent on sensing of viral or bacterial material inside the cells. And this will recruit this ASC protein, which will then bind to procaspase 1, which will activate itself and induce two things: cell death and the release of IL-1 and IL-18. So it's pro-inflammatory. We've got these three mechanisms of cell death, okay? And how are they activated by virus infection? Okay, so all of this detection of viral infection is based on the detection of viral nucleic acids. So double-stranded RNA can be detected by the RNA-dependent protein kinase, PKR, and one of its effects is to activate apoptosis by activating caspase 8. Now, other receptors for double-stranded RNA, RIG1 and MDA5, activate ASC and caspase 1, so they will activate pyroptosis, which can also be activated by some double-stranded DNA receptors, AIM2 and IFI16. So IFI16 is very important in the pathology of HIV infection. So in untreated infection, you don't find very many productively infected CD4 cells, but a lot of CD4 cells are killed, they're dying, and that's because they have an abortive infection. So the virus enters CD4 T cells, reverse transcription occurs, so you get double-stranded DNA in the cytosol. This is detected by IFI16, which activates pyroptosis. So all these cells die, and you get inflammation. So that's very important for the destruction of CD4 T cells during infection. So necroptosis is induced by this DNA receptor, DAI, and some double-stranded, well, not really double-stranded DNA receptor, but virus infection can induce apoptosis by activation of P53. So you get the induction of a DNA damage response. P53 will induce some of these pro-apoptotic members of the BCL2 family. So this is how virus infection induces programmed cell death. Okay, so these are the pathways. Now, viruses don't want their infected cells, their host cells, to die. They want their infected, or at least not very quickly, they want infected cells to survive until they can release a lot of progenitor viruses. Okay? So if infected cells die too quickly, this is bad for virus replication. So many viruses inhibit different steps of the apoptosis pathway. And in fact, herpes viruses also inhibit the necroptosis pathway. So this is necroptosis. This is the backup, right? If apoptosis is inhibited, cells should still die by necroptosis. But herpes viruses, infected cells, don't do that because they prevent necroptosis. Now, as far as I know, there are no specific virus inhibitors of pyroptosis. But probably there are some, and we just haven't discovered them yet. Now have a look at these viruses here, adenovirus, EBV, herpes simplex, pox virus. What have they all got in common? Yeah, especially you should know this. Intern in virology. If I added polio virus, that would be the odd one out. Why? Hepatitis C virus doesn't do it. Why? What do all of these viruses have in common, and polio virus, hepatitis C does not have in common? Does not share that characteristic. Anybody in Ren? You can answer too. It's all gone quiet in Ren, hasn't it? They've turned their microphone off, I bet. No? Okay, anybody, any idea? They're all DNA viruses. Okay, so for reasons I don't know, and nobody knows, only DNA viruses seem to care about inhibiting apoptosis or programmed cell death, whereas DNA and RNA viruses have got a lot of accessory proteins that inhibit the interferon system and these restriction factors. Okay. Right, so the time has come for me to have a little break, and I'm going to send out on a little question to you. Okay, so I'm going to send out a question to you on the internet and see if you can see if you've been following what I've been talking about. So here's the question. So if you have a connected telephone, you've got a smartphone, or if your computers are connected, then you can log on to this site. And the session key is, everybody listening, the session key is 226737. And send your answers in, and we'll see if everybody has understood what I've been saying. Two 737. Let me just check. Okay, there it is. What I need is a little file with some like music I can play during this time when I'm waiting for the answers to come through. Okay, I've got some answers, so let's have a look and see what you've been sending in. Okay. All right, so here's what we got. So restriction is a dominant phenotype. Only 2 people out of 5 said that. That's the correct answer. Yes. Okay. That's dominant. That's the whole basis of starting out the cloning strategy. If you fuse a non-permissive and a permissive cell, if the fusion is non-permissive, that means it's a restriction factor. It's dominant phenotype. Restriction factors are strongly expressed in a constitutive state. That's false. So most people got that right. Number 3, yeah, each restriction factor can target multiple viruses. That's true. They're not specific for one virus species. Maybe on one virus class. Okay, so there might be some that are specific for retroviruses, but they're not specific for one species. 4, each virus can be targeted by several restriction factors. True. And then this one, number 5, last one, RNA viruses are more sensitive to restriction factors than DNA viruses. I'm not sure that that is true, actually. I think probably restriction factors are effective against DNA and RNA viruses. But most of the ones that we know have been identified by looking at HIV or RNA. No, what am I saying? HIV or influenza virus. So the viruses with an RNA genome are at least in the particle. So up to now, most of the ones we know perhaps are more active against RNA viruses. But that might just be an artifact of how we've been looking. Okay. So thanks for that. So we only got 40 minutes left, right? So I better get moving. So we're going to start looking at the adaptive. Wow, okay. First the cellular response, innate and then adaptive, and then the antibody response. Okay, so what's the point of the cytotoxic cells? Well, if infected cells don't actually kill themselves or don't block virus replication by a restriction factor, then CTLs and NK cells can come along and kill the infected cells and prevent spread of virus in the organism. So that's the objective here. Right, NK cells first. How do they recognize cells and decide whether to kill them or not? So NK cells express a whole bunch of NK cell receptors. Some of them are inhibitory, some of them are activating receptors. And whether this NK cell becomes activated and will kill the target depends on the balance of signals from ligands for the inhibitory receptors and ligands for the activating receptors. So normally, a non-infected cell expresses HLA class 1. This will bind KIR inhibitory receptors, and this will prevent this cell from killing the non-infected cell. So if you've got virus infection, one of the things that happens is that very often HLA class 1 will be downregulated to try and escape from the CTL response. And part of the innate response to infection is increasing the expression of these MIC-A and MIC-B proteins that are ligands for activator receptors. So now this NK cell is going to become activated. So you've got a strong activating signal and a weak inhibitory signal. So this cell will kill the infected target cell. So also there's a link here between the interferon response. So NK cells are going to become more active and start to proliferate in response to interferon. Now viruses don't like their infected cells to be killed. So many viruses express proteins that are going to inhibit the activation of NK cells. So cytomegalovirus, it's a herpes virus. This has got proteins that are going to inhibit the expression of MIC-A and MIC-B on the surface of the infected cell, and that's going to stop NK cells from killing cells infected by CMV. Oh yeah, I forgot about that. So they will downregulate the endogenous HLA class 1, and so they'll replace that with a kind of viral version of HLA class 1. So these cells that are infected by CMV will have a low level of activator ligands and will have a high level of inhibitory ligands. So they escape from NK cell lysis. Now for the CTL response, okay, so you have viruses that infect the cell. They'll start expressing their proteins. They can be processed by the proteasome, transported into the endoplasmic reticulum by the TAP complex, and then loaded onto HLA class 1 and then exported to the surface, where CTL can see this peptide HLA class 1 complex. Now, this is going to be useful to eliminate infected cells if you target virus proteins that are expressed very early after infection. So an effective CTL response is going to be targeting early proteins of the virus or capsid proteins. That's a bit unusual because capsid proteins, they're structural proteins, they're expressed late during the replication cycle. But it is effective because capsids that are incoming into the cell, many of them can be destroyed by the proteasome. And peptides from the incoming capsid can be displayed on the outside of the cell. So these are going to be the very, very first peptides that are expressed on the outside of the cell or displayed on the outside of the cell. So if you have a CTL that's specific against a capsid protein, that can be very, very effective at inhibiting viral replication because this infected cell can be infected it can be killed very, very quickly by the CTL, maybe even before expression of viral genes. So CTL that target early proteins or capsid proteins, these are the ones that have the highest antiviral activity in vivo. Okay, so lots of CTL, lots of viruses want to escape from CTL. Once again, they don't want infected cells to be killed too quickly. And there are really two kinds of mechanisms by which viruses escape from CTL lysis. So RNA viruses, like hepatitis C, and retroviruses, like HIV, have a high mutation rate and a very high viral load. So they can generate a lot of mutants, and some of these mutants can escape from CTL, either because they avoid proteasomal processing, or they can prevent TAP transport, or they can prevent the processing of the epitope in the endoplasmic reticulum. Some mutations can prevent binding of the epitope to the HLA class 1 molecule, and some of them can prevent interactions between the peptide HLA complex and the TCR of the CTL. Okay, so mutations in or around CTL epitopes can allow these viruses to escape from CTL. Okay. The other mechanism, which is used by DNA viruses, is, okay, these guys, they don't have such a high mutation rate. But what they can do is they can express proteins that will inhibit different steps of the peptide processing. And they'll prevent virus peptides from being expressed on the surface of the cell. So herpes viruses express proteins that are going to prevent viral peptides from getting onto the surface of the cell. And so that's how they escape from the CTL response. Okay, so that's the kind of interaction between the host response and the viral defense here. So what's the role of this cytotoxic response during a natural infection? Okay, so this has been studied in a lot of models of infection and in real human infections. So first you get the NK response 2 to 4 days after infection. Then you get CTL after about a week. And if you have an effective CTL response early during an acute infection, this is very good. It's a factor for a successful resolution. So an example from like clinical human infection is that in measles, the cutaneous rash that is provoked by the infection, this is the CTL coming into the skin, killing the infected cells. And if you don't see the rash develop, this is a sign of a very poor prognosis. So if you don't have the CTL response in an acute infection, this is very bad for controlling viral replication. So their role in acute infections is really to control the virus. Now in persistent infections that can either be latent or chronic, CTL still have a role. So for chronic infections, HIV, hepatitis C, hepatitis B, okay, CTL are not 100% effective. They can never eliminate the infection, but they do keep the viral load down a little bit. Okay, so people with some favorable HLA class 1 genotypes, they have a lower viral load. They control their virus more effectively. And in latent infections, CTL are preventing reactivation episodes. So can we use CTL, in the clinic? So in fact, yes. So there have been some studies on adopted transfer of CTL lines into patients who have had a hematopoietic stem cell graft. So during the phase where these people get the graft and before they reconstitute their bone marrow, they're very, very susceptible to virus infections. Things like Epstein-Barr virus, adenovirus, BK virus. They're normally kept in check by an active immune system. So this is a clinical trial published last year on using CTL to treat patients who have got these viral infections after hematopoietic stem cell graft. So here's a couple of results. This is a person who's got 2 viruses replicating actively, BK virus and Epstein-Barr virus. In the dotted line, this is the viral load. This line here is the amount of virus-specific T cells. And this is the moment when they transfer the T cells into the person. So quite rapidly after the transfer of the T cells, you get the viral load disappearing. And the T cells divide inside the person. And so they are getting activated by the virus that's present. And they control the viral infection. So that's quite successful. Another way of looking at this is in this patient who had a post-transplant lymphoproliferative disease caused by Epstein-Barr virus replication. So on the CT scan here, you can see these lymph nodes that have got lesions here caused by the cells that were infected. They're proliferating. So this is before the T cell therapy. This is afterwards. It's all cleared up. So in terms of clinical disease and in viral load, this therapy seemed to be effective. So CTL seem to be effective for controlling viral replication, and perhaps we can use that in therapeutic context. Can they actually prevent viral infection? Can we use them in a preventive strategy? So there's been a lot of work in mouse models on this, going back even 50 years practically. And if you perform adoptive transfer experiments of CTL, one animal that's been infected develops CTL. You take those CTL, you put them into a naive animal. Are these animals protected from infection? And the answer is yes. Okay, so there's a lot of data from mouse models for different viruses that shows that when CTL are present, they can prevent primary infection. And in mouse models as well, this also works by vaccination. So PP89, okay, it's an immediate early protein of a mouse cytomegalovirus. So if you vaccinate mice with this protein, they develop CTL. They don't develop a useful antibody response. And these mice are protected against an infection. So judging by these animal models, it seems like CTL can prevent primary infection. So does it work in humans in terms of vaccination strategies? And really the big test of this was the STEP trial, which was financed and developed by Merck. It was conducted from 2004 to 2007, so it's a phase 2B trial looking at efficacy in about 700 vaccine recipients, 700 placebo vaccine recipients. So the idea was, okay, we'll give them the vaccine and we want it to induce CTL and prevent infection in the patients who received the real vaccine. So 73% of the people volunteers who got the real vaccine, they developed specific CTL. So it was effective in this respect. Biologically, there was a response. But in terms of preventing infection, in the group who got placebo, 21 infections. In the group who got real vaccine, 24 infections. 0 efficacy. All right. So in terms of human vaccines, CTL do not seem to be working. All right, or at least up till now. And one of the reasons might be because in the mouse experiments, you choose when the challenge occurs. So you vaccinate, you get CTL, and then you then you infect the animals. So the challenge occurs when the CTL are still around. Whereas in a human vaccination context, you vaccinate, you have CTL for perhaps a short time, and then the infection will occur, who knows when, maybe a lot later. So this could be the big difference, why this approach seems to be effective in animal models. This trial was done based on, encouraging data from the macaque SIV model. So in a primate model, it also works, but in humans, not so good. Anyway, so I think the big thing is that in the model, you control when the infection occurs, and in a real situation, you don't. Okay. We've got about 25 minutes left. Do you want another quick clicker question or not? You say, yeah, okay for a clicker question? Yeah? Okay. Right, so I'm going to send out this question, and it will be a different code number. If I can. Okay, so the code is 610065. Okay, there it is there. Yeah, I think you have to think about these questions a bit more, perhaps, than the previous ones. Okay, it's taking a bit of time to get answers, so let's just look at the 3 answers I've got already. That's it. Maybe the other ones will come up. Okay, so that's what we've got sent in so far. So CTR less important than antibodies in the resolution of infection. That's false. Okay, everybody got that. Cells that are infected that inhibit apoptosis can't be killed. That's also false. Everybody got that right. DNA viruses can't escape from a CTR response. That's also false. They express proteins that inhibit the steps in processing and presentation. The initiation of a CTL response depends on the activation of an innate response. That's true. Okay, CTL need dendritic cell activation. Otherwise, the specific response can't get going. So that's true, everybody. Okay, 3 people got that right. This last one. NK cells recognize viral peptides on the surface of infected cells presented on HLA class 1 molecules. That's not true. That's false. Everybody got that wrong. CTL recognize cells like this, infected cells like this. NK cells, they just recognize these activator ligands, inhibitory ligands. So NK cells is MIC-A, MIC-B that's going to activate the activator ligand. Okay, they don't directly recognize HLA and the virus peptide. They recognize rather that there's not very much HLA on the surface of the cell. So you 3 people, that's wrong. Okay, at least I'm happy when people don't get all the answers right, because it makes me think the whole thing wasn't a waste of time. So, right, so quickly, we have about 20 minutes to cover the antiviral humoral response. So what is important here is neutralizing antibodies. Okay, so neutralizing antibodies are the ones that bind on the surface of the virion, the virus particle, and they'll block either the interactions between the glycoprotein, the attachment glycoprotein, or the attachment protein on the capsid, and the cellular receptor or the co-receptor. And neutralizing antibodies can also be active by blocking fusion between the virus envelope and the cell membrane. Now, neutralizing antibodies are important for memory and long-term resistance because they can be secreted into the mucosa. So they can stop virus infection at the very first cell that's going to be infected. So that's why the humoral response is important for sterilizing immunity. So visually, there's something like this. So this is a rhinovirus particle with its receptor ICAM1. The neutralizing antibody binds at exactly the same site on the capsid that interacts with the receptor. So that's why if you get these antibodies bound to the particle, then this particle can't interact with the receptor anymore. So one thing you need to remember is that not all antibodies are neutralizing. So you can have somebody that is seropositive on an ELISA or a Western blot, but maybe their neutralizing response is no good, and they can still be susceptible to a particular strain of virus. Now, okay, neutralizing antibodies, they're induced when you get successful resolution of an infection. So can we use them in therapy? So yes, okay, so for a long time, serotherapy with the gamma globulin fraction of a hyperimmune serum has been used as a kind of a post-exposure prophylaxis against hepatitis B in babies, rabies virus, rabies infection in people who've been bitten, and respiratory syncytial virus in, once again, young children or babies who are very susceptible to RSV infection. So babies that are premature will be highly susceptible to severe RSV infection and can be preventively treated by gamma globulin serotherapy. So this is kind of being replaced now by using monoclonal antibodies. So for RSV, there is a product that can be given to, it's used in pediatric medicine. For rabies post-exposure prophylaxis, CL184, this is a combination of two monoclonal antibodies that is given instead of like the hyperimmune serum from animals. And ZMapp, this is the one that's it's a cocktail of monoclonal antibodies used successfully to treat people for Ebola virus infection. So using neutralizing monoclonal antibodies, this is a successful therapeutic strategy. I'm going to skip the next two slides about the development of the preclinical development of the ZMapp cocktail because it's a little bit less topical than it was last year. And let's go, and I just want to use the time that I have left thinking about neutralizing antibodies and vaccination. Okay. So the reason why we have antiviral vaccines that work against some viruses and vaccines that don't work so well against other viruses is to do with the neutralizing serotypes. So whenever you've got one serotype or a limited number of serotypes, then we've got a pretty effective vaccine available. Okay, so you vaccinate against poliovirus with the 3 serotypes, and that's it, you'll be protected. Now, another case where vaccination fails is for rhinovirus and adenovirus. So these viruses circulate, and they have like 50 or 100 different neutralizing serotypes. So there's no way you can vaccinate against all the viruses that are in circulation, that are circulating in the human population. So it is possible for these viruses to produce a vaccine that's got a kind of limited range that protects you against a limited number of serotypes. So, for example, the US Army for a while vaccinated people against particular adenovirus serotypes because they'd had problems in their barracks with epidemics of one particular adenovirus serotype. It's not used anymore, but they tried it out in the 1990s. One that works for this is papillomavirus, HPV vaccine. There are a lot of different HPV serotypes, but there are only 2 very high-risk HPV serotypes for cervical cancer. So you can vaccinate successfully against particular serotypes. Okay? Now, another kind of problem is when the serotypes are variable over time. That's particularly true for influenza. Okay, the virus is not stable over time. So the vaccine has got to change and be produced every year. So we don't have a universal vaccine available, but we have vaccines that are produced every year that are effective against different serotypes. So if the virus mutates over the year, then the current year's flu vaccine can be ineffective. And so trying to get a universal influenza vaccine is a big problem in vaccinology. So those are the kind of like ordinary problems for serotypes and neutralizing antibodies. There are some viruses that don't really seem to fit into this like tidy classification. So HIV will be one of those, and respiratory syncytial virus would also be another case where we don't have a vaccine available, and it's not really due to the number of serotypes. Okay, so let's have a look at HIV. Why don't we have a good vaccine available for HIV, all right? And can we actually get neutralizing antibodies against HIV? So this is data from a study published in 2003, which I think was really nice, because what they did was they isolated plasma at different times from this person after their initial primary infection. 0, 3, 6, up to 25 months. And then they got the virus that was circulating at these different time points. So they were able to test the neutralizing activity of the serum at each time point against the viruses that were circulating. Okay, so at like month 0, primary infection, you don't have seroconversion yet. Okay, so this serum doesn't neutralize any of these viruses. Okay, that's normal. 3 months, what have you got? Okay. The serum has a weak neutralizing titer against the initial virus and the virus that's circulating. At 6 months, you've got a good neutralizing titer against the initial virus and the virus that was circulating 3 months before. But there's no neutralization of the virus that's actually there at that time. And if you go down along the diagonal here, you'll see that at every time point, the neutralizing titer is really pretty low against the virus that is actually circulating at that time. So what is happening is that neutralizing antibodies are being generated in this person, but the virus is escaping from them. The virus is mutating and it escapes from the neutralizing antibody response. And that's why the virus that's around is always not getting neutralized and can go on and infect other cells. So the lesson from this is that people who are infected by HIV, they do develop neutralizing antibodies, but mostly they are very limited activity. So they'll neutralize some particular viruses, but they won't neutralize a broad spectrum of HIV. So this is why the virus continually escapes from the humoral response, and that's why we haven't got a good. Well, okay, well, I'm selling the punchline a little bit. Okay, so some people do develop broadly neutralizing antibodies. Okay, so this is the case in most people. Most people don't develop broadly neutralizing antibodies, but some people do. And these very rare antibodies have been studied. Okay, so what are the steps in HIV attachment and entry? So you've got GP120 will bind to the receptor CD4. This induces a conformational change, and the V3 loop will bind to the co-receptor CCR5, and that allows deployment of the fusion domain, which is in GP41. So if antibodies can block any of these processes, then infection will be prevented. Okay, so neutralizing antibodies can block any of these steps. Now the V3 loop, V is for variable, right? So that's not going to be very broadly neutralizing response. So this one, interaction between the V3 loop and the co-receptor, that's not the target of a broadly neutralizing response. However, so all these V3 loop antibodies, they're going to be very narrow specificity. The ones that are broadly neutralizing, they can bind the CD4 binding site because this is going to be quite highly conserved. And another class of broadly neutralizing antibodies bind to GP41 and they'll prevent fusion. So if we could design a vaccine that could induce these broadly neutralizing antibodies, then it might actually work and be an effective vaccine. So do we have it yet? So these are the results from the biggest phase 3 vaccine trial in HIV virology. So these results were published in 2009. This was a randomized trial. So we've got 16,000 people. 8,000 get placebo, 8,000 get the active vaccine. It was partially protective. This was a better result than the STEP trial from Merck. So, you know, about 30% of the infections were prevented. But it doesn't seem to be due to the induction of broadly neutralizing antibodies. So there's some other mechanism involved here. And it's not clear yet whether this vaccine can be modified in order to be able to permit it to induce broadly neutralizing antibodies, or whether it's just going to be a dead end. We don't know that yet. So there are a couple of big groups in the States looking at ways to modify the structure of GP120. Because what the observation is, in GP120, when you use it to vaccinate, it will induce some neutralizing antibodies but have a very limited range. It doesn't really induce broadly neutralizing antibodies. And so one of the strategies is to try and modify the structure of the immunogen so that it will be able to generate some broadly neutralizing antibodies. And if they can do that, then maybe that could work. And there's another approach, very, very different from Morgan Bumsed in Cochin. So her idea is to try and forget about this CD4 binding site. Try and target the GP41 and get antibodies that can prevent the fusion of the virus particle with the host cell. And so this is another kind of immunogen. Seems to work, and the clever thing about it is it's a prime boost strategy. So you get the first injection intramuscularly, and the boost is intranasally. And that can induce IgA at the mucosa, because that's what you want. You want to have IgA in the vaginal mucosa to prevent sexual transmission. So this has been effective in macaques, in a macaque model, and the phase 1 study was published in 2013, 2014. So I don't know how far this is going on its development. Okay, time is up more or less. So the main points to remember here is, okay, how does a cell know whether it's infected? So these are toll-like receptors on DC and the intracytoplasmic receptors for different viral nucleic acids. What do you do if you're infected? Induce the interferon response or induce programmed cell death. And then in terms of the immune response, okay, for the cellular response, how do NK cells and CTL recognize infected cells? And for the humoral response, what are the characteristics of neutralizing antibodies? How can we use them in therapy and in preventive strategies by vaccination? Okay, thanks very much for listening. Sorry I was a bit rushed at the end. Are there any questions? Nope. Okay, so I will not see you tomorrow. I'm not coming to Rennes, but I hope you enjoy it. Okay, so see you next week by Visio, I guess.